Hello, everybody, and welcome to this week's webinar. We will go ahead and get started in a couple of minutes. People are still joining. While we are waiting, as customary, we usually ask people to go ahead and fill up this questionnaire. It's a quick question asking which database you use. You can go ahead and type this URL into your browser, or you can scan the QR code. I will paste the URL also in the chat box, so you can also click on it from the chat box. And once you get there, you will see something like this, and I will go ahead and start the poll. So you can start answering, and you can let us know which database you use. And this is a multi-select, so you can select more than one database. So we'll let this go for a couple of minutes as people continue to join the webinar. And again, if you're just joining the webinar, we're quickly doing the online poll to see what database you use. The link is in the chat, or you can scan this QR code using your phone, and that should take you straight to the poll. I'll leave this up for a couple of seconds while we're waiting. Also, while we're waiting, in your control panel, in your GoToWebinar control panel, there's a question section, which allows you to go ahead and type questions you have during the webinar. We have a number of ViewPathDB staff members who are on the call as well, and they will answer the questions as the webinar is going along. Or we may answer it verbally using a demo in case that's required or needed. Also, the webinar is being recorded. And so if you miss anything, or if you want to share it with your colleagues later on, you will get a link to a YouTube recording of this webinar. So if you miss something, don't worry, you'll have access to it later on. And you'll get the email probably tomorrow sometime, although the YouTube might be up on our YouTube channel later today. Okay, also as we're waiting and people are still voting, if you're curious about other webinars we hold, if you go to any of the ViewPathDB websites, so viewpathdb.org, you can go under the help menu, click on learn how to use ViewPathDB, and you'll notice here there are multiple buttons that you can select. One of them is webinars, and if you explore these webinars, you will get a list of upcoming webinars. There are a few more that we're still scheduling and more to come, obviously. But you will see that next week is a webinar on building complex strategies. And so we'll go through building what we could like to call in silico experiments. So ways to combine different types of data together. And so that's very useful. There's another webinar following that on August 20th, which is all about metabolic pathways in ViewPathDB. So you'll be able to learn how to find various metabolic pathways, find genes that are involved in these pathways, and also paint these pathways with different experiments and phylogenetic profiles. So that's it's actually a pretty cool feature, and I would encourage people to attend as well. And of course, if you've missed any of our previous webinars, they are all listed on this page. And each of them, if you click on any of these, it'll take you to a section on the page that includes a short description and a link to the YouTube channel or the YouTube video itself, and also any handouts that were used during the webinar. Okay, I think we are reaching a good number of attendees. Let's take a look quickly at the direct poll. Looks like we have a good number of FungiDB users. I'll keep this poll open throughout the webinar, so you can feel free to answer them throughout the webinar. Also, we usually get requests from people about certificates for the webinar. You'll automatically get one tomorrow when you receive the email from GoToWebinar that includes the link to the YouTube tutorial. All right, so without further ado, we're going to go ahead and get started. So today's webinar will focus on HostDB. So HostDB is one of the ViewPathDB components, and the goal of HostDB is to provide you with access to host response data to pathogen infection. Right, so if you have you often think about the parasite and or the pathogen and what is changing in the pathogen as it's infecting or going through either interacting with a host cell or entering a host cell. And obviously the parasite goes through many changes, but of course the host also goes through many changes. And more where people are actually, when they're working on an infection model, the data they collect, for example, RNA sequence data or proteomics data, actually, unless they specifically purified parasites, the data, the samples they're collecting contains both data from both the host and the parasite. And so if you do an RNA sequence analysis of the RNA you're getting from an infection of let's say a macrophage with toxoplasma, you are going to be getting RNA sequence results from both the host and the parasite. And so you can ignore the host RNA sequence data if you want and just look at the parasite, or you can look at both. And more obviously people are looking at both because you're able to collect now sufficient data from both the host and the parasite to actually analyze it and find some meaningful results. And so HostDB, the goal of HostDB is to start integrating the host response data, and again, to provide you with all the tools that are available in ViewPathDB, similar to if you're familiar with FungiDB, for example, you'll be familiar with the searches there. And so the goal of HostDB is to provide you with these searches so you can start now interrogating the host response data to infection. What we're going to do in this webinar, there'll be just a couple of quick things. So after this quick general intro, we're going to describe some of the available searches. And I'm joined by Evelina Bashenko, who's based at the University of Liverpool, and Suzanne Warrenfels, who's based at the University of Georgia. I'm Omar Harb, based at the University of Pennsylvania. So both Suzanne and Eve are going to take you through a few example searches and types of searches in HostDB. And then we're going to finish up by, if we have time, I'll run another example search where you're integrating between different experiments to see if you can find some interesting correlations between infection models. So HostDB, again, if you're not familiar with ViewPathDB sites, you will notice that, or if you are familiar, then you'll notice that this looks pretty much the same as any of the other ViewPathDB sites. So if I go back to viewpathdb.org, you'll notice that the layout is pretty much the same. The colors are slightly different, the different organisms up here at the top. But the bottom line is that this layout hopefully is intuitive. You have a banner at the top here, or a header section, which gives you quick access to the searches, different tools on the database, your workspace. So this is a place where you can analyze your own data. Data downloads, so all the data that's available in the resource, we provide it to you for download. We don't provide for you the RNA sequence data because there are repositories for that. So for example, if there's an RNA sequence experiment and you're interested in getting the raw FASTQ files, you can actually, we provide you information about where you can get it from, but then you can go and get it from the Sequence Read archive, for example. There's information about this resource. Help, as I mentioned before, there are different good sections, but I would again recommend that you go to the Learn How to ViewPathDB, to use ViewPathDB section, because there's a lot of useful things there. The Contact Us link is really important. It allows you to communicate with us. So if you ever have a question on any of our resources, you can click on the Contact Us link. This just takes you to a quick form, which you can fill up and send it. And a number of us will get the email, and we try and respond to you within 48 hours. So, and know, you can ask us anything from, how do I find a particular search, to something that's more complex and you're trying to do some analysis and you're just not sure where to go. And if we have, obviously we have the answer, we'll give it to you. If not, we'll try and point you in the right direction. There's links to our social media up here. And also importantly, we have this ability to log in. You don't have to log in or register and log in to use HostDB or any of our resources, but there are some features that require you to log in because you're then storing information. So for example, if you want to analyze your own data in a private workspace, you have to create a login. Or if you want to add comments on genes, right? These are public comments on genes, but they're associated with you. You can do that, but you have to log in and create a registration. It's a really simple task. All we ask from you is an email, your institution, some just very, very basic information. You can sign up for newsletters if you're interested in hearing about what's new in our resources and special events, and you register, and then you automatically get a password, which allows you to then log into the resource. All right. Going down here to the middle section, you will notice that there's a section here on the left called Search For. One thing about the header, you'll notice there's a big search box up here. That's our site search, and we will demo this in a few seconds. But on the left here, there are some specialized searches. So you can search for genes, for example. You can search for organisms. You can search for genomic sequences. And you will notice that each of these can be expanded into additional submenus. And of course, these categories can be bewildering. You're thinking, oh, I have no idea. I'm interested in secreted proteins, so I'm looking for a search for signal peptide, but where will I find it, right? And so you could try and go through and figure out, well, maybe they meant it's under protein target localization. That seems logical, but maybe it should be under protein features. But to take away the guesswork, we provided you with this filter here. So filter the searches below. So I can start typing signal right here, and immediately it filters my searches, and I can go to the predicted signal peptide search. So I highly recommend that you play around with this filter option here and find searches you're interested in. But you'll see here the broad categories. So you're in this particular section, you're specifically searching for items that fall under these different categories, and they all have specific searches that are available to you, like the signal peptide search that I just showed you. The central portion here includes a lot more help. So all of these little buttons here will give you some little vignettes on what you can do in the database. So for example, if you're interested in, well, what are search strategies? You can click on this and you will find that there's a little bit more information about search strategies to get you started. If you want to interested in what kind of transcriptomic resources are available, if you are interested in downloading data, right? These are all little vignettes that will walk you through a step-by-step process on how to quickly get started. And if you scroll down to the bottom here, you'll notice there's some more extended tutorials and exercises. So this will take you to a PDF, which takes you through a step-by-step exercise where you can follow it step-by-step, and it'll teach you about how to, for example, run site search. Since all of our databases are built using the same infrastructure, you might go to site search, and this may be using vector-based, for example, as the example, but the same things you're doing in that search will work anywhere else because it's the same functionality, basically. And the last thing I want to say about the homepage and the layout before I turn it over to Suzanne and Eve to start talking about specific searches is this news and tweets section. So if you expand this section, you can access our most recent news releases. So if you're interested in knowing, well, what happened in release 47 or 46, you can go in here and click on these and get more information. We also have a stream of our Twitter feed, and so that's a nice way if you're not on Twitter but you just want to see every now and then what's been tweeted on ViewPathDB, you can quickly take a look at this and learn more about what's going on. And you can always collapse this section right here. Okay, I think this is all I wanted to say about the introduction. So I will go ahead and switch it over. I believe I am switching it over to Suzanne. I think you're taking up the next step.
Mm-hmm.
Great. So let me make you the presenter.
Oh, is that true, Eve? Were you? Eve was going to do site search.
Yeah, let's start with site search and then move on to search strategies.
Sounds good.
All right.
All right, you should be the presenter now.
All right, sounds good. You should be able to see my screen now.
Yes.
Yes.
All right. So the next section, Suzanne and I will tag team. I will introduce you into site search and Suzanne will take you into the details of how to create search strategies. And later on, we will also demo a few specific search strategies using the integrated data sets. So as Amara has mentioned, we have several sections to the website, and the site search can be deployed from this bar right here at the top within the banner of HostDB website. So site search is located at the top. So even if you navigate away from the main landing page and create search strategies, it will be still displayed for you, so you can access it at any time. And using site search, you can search, for example, for a gene ID, if you know a specific gene ID that you would like to take a look at. Or you can also search for a text or a text term. And essentially, your results will include all records or terms that include your search entry. So let's give it a try. I will create a search for cytokine and click enter. I misspelled it, so that will help if you actually spell it correctly. Here we are. So notice that once the page is refreshed, you essentially have a number of results available to you. And the page consists of several panels. First, you have a panel on the left that essentially summarizes all of the records that we have that have the word cytokine mentioned. You also have sort of the main page or the center of the page that lists all of these results. And there's also a button here on the right. And as we go through setting up our search a little bit further, watch what happens to this button here. So let's take a look at our results. My search returned records for genes and PopSet isolated sequences, and I would like to go ahead and select one of the filters and would like to identify all genes that essentially have the mentioning of cytokine in either Go terms or Inipro domains or product descriptions. And I will click apply to confirm these selections. Notice that while I was doing these selections, the button on the right has changed the color. Now it's bright blue. And that essentially means that now your results are ready to be exported as a first step for this search strategy. And so before we move on and actually create a search strategy, I will go ahead and define that in addition to all of the selected terms here, I also would like to look for these records, let's say in a mouse. So you can either expand the available menus on the left, or you can also search and bring up an organism here. I'll go ahead and apply. Okay, so now I'm set to look for the genes that mention cytokine in these particular terms and also within the mouse genome. And go ahead and export as a search strategy. So notice now that you have essentially in a named search strategy that you can change the text within each step to help you remember, for example, what you have searched for, such as I'll say here I have searched for cytokine. And this is your starting point to essentially create a multi-layered search approach to interrogate the integrated data in HostDB and also your personal data, let's say if you're importing them from Galaxy. And check out our previous webinars for more information on how to do it. And as Suzanne will go in a little bit more detail just in a few seconds, to expand the strategy, all you need to do is click on the Add step and essentially choose from the available options. All right, Suzanne, I will give it back to you.
Here we go. It says you're the presenter. Here we go.
Oh, should we make you? Okay, there you go. Mm-hmm.
Yeah. Okay.
So one thing, maybe before you get started, you noticed when Eve made a typo, right? She misspelled the word and the results were 0, of course, and that's to be expected. So it's one thing to note is that search is not like a Google search where it's going to try and find anything in the world that looks similar to the word that you look for. It's limited. And so that's something, we are looking into potential options of, providing you with like a dictionary almost. So if you mistype a word, it may say, well, did you mean the following words? That's obviously a bit more difficult for scientific terms, it seems like it, but certainly something we're looking into. Anyway, sorry, Suzanne, go ahead.
Okay, yeah, no problem. So yeah, Eve has shown you how to use the site search then to explore really where things are, and you can also use that to enter the strategy system. But I'm going to do a similar text search, but use these specialized searches that we have. That
Suzanne, we're seeing your second screen, I think. So we're seeing.
Oh my goodness.
A beautiful picture of somebody on a canoe.
Yeah, I guess I should do this.
There you go, it's coming in there. Okay, perfect.
Yeah, I'm sorry.
No problem.
Okay, so I'm at HostDB and I want to demonstrate the strategy system, right? It is integrated with the site search, but you don't have to start with the site search to use the strategy system. And when you use ViewPathDB, you have at your fingertips a wide array of pre-analyzed omics data. As Omar shown, you can access searches here or through the search menu up here. We analyze hundreds of omics data sets according to workflows, standardized workflows, and assemble, our database with these omics analysis results. And then each search then accesses and queries a different set of omics analysis results. And so our database spans many organisms, right? All of the organisms that we've integrated, and many data types, RNA sequence, proteomics, there's variation data, and then many experiments within that, right? So you can imagine that as a computer scientist, we can build a script to pull out related features from our set of tables that spans this wide array of organisms and data types and experiments. So for example, we can write a script to pull out all genes across organisms that are expressed at a certain life cycle stage through Rna-Seq data, or that can act as kinases, or that have a specific protein domain. If we have the omics data to support it, we can build a script to pull it out for you. And that's and that's what we do with our search strategy system. We have developed a set of queries, over 100 queries, and then we build an interface for you to interact with these queries and run these queries without having to do like command line stuff. And then, the superpower of our strategy system is that you can string these searches together to find subsets based on other omics data, right? So you can say, show me all genes that are likely kinases and that have transmembrane domains. In general, you can use our strategy system to find a set of genes that share biological characteristics that you are interested in. So that's background, and I'd like to go ahead and demo that. I wonder if there's any questions.
No questions so far. So again,
Okay.
If people have questions, go ahead and type them in the question panel, and we will answer them as they come along.
Okay, so I'll get started on our strategy, and the example that I'm going to use today is to look for secreted kinases. So, and to do that, we're going to build a 3-step strategy, and that we'll use the text search to look for kinases, and then we'll use another search that accesses the analysis results of our signal P analysis, which predicts signal peptides, which indicate that a gene's protein product would be targeted for secretion. And then we'll run another search for transmembrane domains, which is based on our analysis of whether a gene is predicted to have a transmembrane domain or not. Okay, so let's get started on that. I am at the home page of HostDB, and I'm going to run a text search for the word kinase, and I'll find that search over here. I want a text search that returns genes, and I'll click that. And then that takes me to the search page with the title at the top, and then several parameters. We have organism, the text term, a fields parameter, and a get answer button. So it's a little bit different than the site search, running a text query from the site search. If I keep scrolling down, then I have some help information, and then links to data that is queried in this search. So let's set this up to look for kinases across all of our organisms. I'm going to choose select all for our organism parameter. The text term that I want to look for is kinase. And then the fields parameter is a way to direct the text search to look in all of the categories that are available. You could look for kinase only in the gene name or product description, but we have all of this available. I'm just going to keep it there. And then I'm going to call this search kinase and click get answer. And what we find is that we have 7,000 genes that are returned by the search and are associated with or have the word kinase somewhere in their record. Results look a little bit different than the site search. On the left down here, I have the organism filter, which if I expand all, will show the distribution of the hits across all of the organisms that I searched. And then over here on the right, I have a couple of tables. I'm looking at the gene results. And this table is a list of all the genes that are returned by the search. The first, or actually second column here, is gene ID, and that links to the gene record page, which you can integrate. And then I have a bunch of other columns that are associated data. And in fact, you can use this add columns tool here to add any data that we have in the database to this table here. Which is great to do, and there's other tricks you can do. You can download this data set and all that kind of stuff. So we could stop here, but what we had said we wanted to do was to look for kinases that are likely secreted. And so I'm going to click this add step button, and now I can choose to add other data to this strategy and look for subsets, right? So I'm going to choose to combine with other genes and intersect my kinase genes with results of another search that looks for signal peptide genes. So I'll look for the search here. I found it. I'll click. And now I am in, now I have another search form that is genes by predicted signal peptide. And this, I'm going to intersect that with my previous search. This only has one parameter here. I'm going to choose select all and then run step. Okay, so then I'm taken to my result page where you can see I now have a two-step strategy. The search that we just ran returned about 31,000 signal peptide containing genes, and 1,400 of them about intersect are in the same list as my list of kinase genes. So these 1,400 genes are genes that are likely kinase and have a signal peptide. And again, you can go down and look at all of that, add more data, and you can even extend this strategy, right? So we also said we wanted to look for transmembrane domains, so I'll click Add Step, combine this with other genes, intersect them with transmembrane, look for that search, and I found the genes by transmembrane domain count search. I'll go ahead and choose all organisms. And in this search, I have 2 more parameters that I need to tell it how many transmembrane domains a gene needs to have before it will be returned by the search. So we've actually set up very broad parameters to begin with, between 1 and 99 transmembrane domains. I'll click Get Run Step. And the search, well, again, returns about 31,000 genes with transmembrane domains. And the combined result is now 623 genes, and these have the biological characteristics that I want. They are likely kinases that have signal peptides and transmembrane domains. So this is a very powerful and actually kind of unique to ViewPathDB, that we have the system where we can take many different types of data and search each one individually, but then string them together. Other tools, you don't always have to intersect. There are other ways to build logic into your strategy and come up with truly a list of genes that you are interested in and have biological characteristics that you want. I thought I'd stop there. Mm-hmm.
Yeah, one thing that would be nice to show, Suzanne, is if you go, for example, hover over the kinase step and select the edit option.
Oh, right. Mm-hmm.
And then revise it. And so the idea here is that when you build a strategy like this, is you can always go back and revise any of the steps, because it's like an experiment, right? You may decide, oh, in a experiment in the lab, you may decide after you look at the results that you need to add a little bit more salt to your buffer because the enzyme digestion wasn't complete, or you need to increase the temperature or decrease the temperature. And the same thing here, you can go back to any step and modify it, change the parameters, and then come back and look at what the results look like, because maybe you weren't finding the exact results you were interested in, or they didn't make sense. And as Suzanne showed you, she went ahead and revised that kinase step and selected different fields to look under. So only now looking in the gene name and description, and then now your numbers are different.
Yep. Yeah.
Great. And one thing also to highlight here is that if you log in, so I mentioned that you do not have to log in to run the search that Suzanne ran right here. But if Suzanne wants to share the search with you, and she wants to say, well, here's the search that I ran, go ahead and look at it and see if this is interesting to you. Then she would, if she can do that by logging in, and now that search, she can save it in her profile, and then she can generate a unique URL that she can share. And maybe while we're, as Suzanne is showing us this right here, she's saving it. And then she will go ahead and share it. And maybe you can, Suzanne, maybe you can share it in the chat box, and everybody can, if they want, they can click on it and they can see it in their own windows. And so you see Suzanne clicks on the share button, and you'll see this unique URL that she will copy, and she will put it in the chat box, and everybody on the call can click on it, and it should open the same exact strategy in your window. It'll be in your own workspace. You're not obviously changing or messing around with Suzanne's strategy. Okay, great. So let's go ahead and switch over to Eve. I think you're going to start demoing some specific searches in host response to fungi, I believe. I'll make you the presenter. Okay.
All right, sounds good. Thank you, Omar. All right, so I will demo a search that comes from the recent publication in 2009, where authors have looked at RNA-seq data coming from both host and a pathogen, and essentially looked at the progression of the infection and also how gene expression changes with various stages of infection. So if we look a little bit closer at the data that we are going to take a look very briefly today. We have a host that is a mouse, macrophages, a pathogen, Candida albicans, and we have several time points when the data was collected. There are several types of samples. For example, there are samples where macrophages were infected with live Candida cells, and as such, the transcriptomes were taken both from host and pathogen. There are also infected macrophages with actually dead Candida albicans cells. There are macrophages that were unexposed, and that will serve as a control in our experiment. And there's also macrophages that have been exposed to the pathogen but have remained unaffected. So in the next few steps, I will take a look at one of the samples and actually across several time points and look for those genes that are upregulated in the first several hours of infection within macrophages in response to infection with Candida albicans. So let's go ahead and navigate to the HostDB site and find the data set that I'm interested to demo today to you. So to identify the Rna-Seq evidence, you can simply search the filter at the top and navigate to the Rna-Seq dataset page. If you know the dataset that you would like to take a look at, you just simply click on one of the buttons, and these are the different types of analysis that we made available to interrogate the Rna-Seq evidence. I'm going to search for a Candida dataset and click on the fold change expression. So in the next few minutes, I will spend essentially setting up an in silico experiment where I will identify a control sample, which is my reference sample. I will choose the comparison samples, and in this case, we're interested in macrophages infected with Candida, where Candida albicans cells are live. And we will also look for upregulated genes. So let's go ahead and set this up. First, notice that you have a few things to choose from, including the type of the genes or the type of the response that you're looking for. I would like to identify upregulated genes, and you could potentially look at twofold, but I would like to increase my stringency a little bit to not say I would like to return genes that are upregulated at least fourfold. And then I'll proceed with selecting control sample. So I'll scroll down through available samples and choose macrophages that were unexposed to the pathogen as my control. And then I will select the first 3 time points, 0, 1, and 2. Or you could also choose just 1 and 2 hours to look at the genes that are becoming upregulated in the very early stages of infection, as we know that the pattern of gene upregulation actually changes by the 4 hours. And let's go ahead and click Get Answer. So notice that as Suzanne showed you before, your search returned a list of genes that fit within the criteria that we have selected. So we could potentially collapse the section on the left to make a little bit more space and examine our data. So if you ever forget the type of the criteria, the type of the criteria that you have chosen, you can click on the edit button and look at the parameters setup. So let's go ahead and examine the data and actually figure out, well, how can we analyze further the 1,300 genes that we have returned. So notice that you have a standard table on the bottom that lists gene IDs and also other types of information available, including the FPKM graphs of how this particular gene behaves within the samples that we have looked at, but also all other samples belonging to this particular data set. You can choose, for example, if you're not interested in displaying some of the columns, you can just close them off, but all columns can be brought back by clicking on the Add Columns button. Additionally, what you can do with this data, if you would like to dig a little bit deeper, you could click on the Analyze Results tab at the top here. And we have several ways to help you enrich your data for various terms. For example, if you click on the Enrich My Data using Gene Ontology Enrichment process, this data will, this search will essentially take in all of the Go terms that are associated with the genes returned in the first step here and look for, let's say, certain characteristics or certain terms that are enriched in your specific result. So let's go ahead and click on enrichment using a molecular function. Notice that you have 3 different types of enrichment. You could select computed or curated evidence, and you also have an option to deploy or not GoSlim terms. I will leave everything at default and just click on the submit button. And while our results are running, here we are, actually quite nice. I will scroll down to the results. So at this point, we offer several types of information. First, you can take a look the genes with this term in the background, the genes that specifically correspond to this Go ID in your search. And we also have various types of statistics available to you. And if you'd like to learn more about what this means, actually check out our previous webinar on Go enrichment as we go in details as to what these things mean. But you could potentially sort by various p-values to identify genes that you may be interested in and examine your data a little bit more closely. However, you can also open and summarize your data as a visual representation of your terms using Revigo. And so I will click on Open Revigo button, but I have already pre-run the analysis, so I'll skip the individual steps. But this is actually the data that we have just visualized in the table. And Revigo summarizes the list of the Go terms that I have just shown you in the table. And it uses a clustering algorithm that relies on semantic similarity to essentially create a representative subset of terms and cluster them based on the p-value provided by the user. So for example, blue and green, as seen here, are more significant p-values from our analysis, and it also provides a graphical representation as far as the sizes of the bubbles that visualized that's shown here, and the size indicates the frequency of the Go term in the underlying Go database. So that's one way to analyze your results. Furthermore, if we come back to our search strategy, we can actually take a look and say, well, you know what, previously I was interested in cytokines, and I want to know how many of these genes have that term associated with them. So I could potentially deploy a text search where I will click on the Add step, navigate to the search combined with other genes, and click on text, and use mouse genome to search for cytokines. Notice that I'm interested in searching for cytokines, but I only wrote cytokine. One way to account for possible variations of this word is actually to use a wildcard, to where this term right now will be searching for both cytokine and cytokines. And I will scroll down. I'll just select a few terms that I'm interested in. I would like to identify genes where this term matches product descriptions, notes from the annotators, names and aliases, inner domains, Go terms, gene names and symbols, and let's say gene ID and alternative product descriptions, and click on Run step. So as the step refreshed, notice that we have 20 genes that now are returned as being fit within our search parameters. And notice that while we searched for cytokines, we have a list of genes that actually have designation as interleukins. And that's because cytokines is a fairly large group of proteins that includes interleukins, lymphokines, monokines, and other types of proteins. So this data actually makes sense, but you can not only investigate further to see what specialized what special types of information you can find about every single gene, and you can navigate also to individual gene record pages. For example, if I click on the gene at the top here, I'll be redirected to a gene record page. And the gene record page is essentially an encyclopedia type of page that contains all information that we know about this particular gene in HostDB. And as you can imagine, you can view the transcriptomics data and notice that this transcriptomic view, or the summary, is a summary not only about the specific data set that I have just shown you, but also all the data that we have from other data sets concerning this gene. And you can also view how the gene expression changes compared to other data sets. All right, I think.
Eve, before you
Yes.
move on, so if you still have the Revigo output open, maybe you can click back on that. And the question was, what do the axes represent in the Revigo graph? And if you scroll down a little bit, you see how the y-axis and the x-axis, they say semantic space y, semantic space x, and the question is, well, what do those mean? And if you mouse over where it says interpretation of the coordinate axes, right there, and you'll see here, it gives you a short description of what that means. But basically, it's a way to represent the Go terms that in your list, cluster them based on similarity, based on the groupings that belong to each other. So you have basically a coordinate space on your graph where they can cluster together, basically. But you can get more information on the Revigo page and learn more about it. I don't know if you wanted to add anything else, Eve, to this, or was that sufficient?
No, I think it's sufficient. Anything that clustered here has a similarity and grouped underneath here as a sort of parent term. For example, everything in gray has been essentially collapsed into methyltransferase activity. And there are several publications on Revigo, easily accessible in PubMed, that go way into details not only about this graph, but into additional types of visual representation of the data.
Great, thank you. So we should switch over to Suzanne. And while we're doing that, I'll just answer the next question, which was from Yasid. He's asking, are there available in HostDB proteomics data like host pathogen protein-protein interactions? And then there's a suggestion for a paper, and that's a really good point, and that's something that we need to move towards in HostDB to provide more of the interaction between the host and the pathogen. So all of the data that's available in HostDB, which is the host response to the pathogen, if you go to the database, for example, the fungal data set that Eve showed us, if you go to FungiDB, you can actually query the fungal side of the equation, right, and ask, well, how many genes are changing during infection? And but we don't have a way to, right now, we don't have a way to tie them together, right? So just because a gene is upregulated in the pathogen and one is upregulated in the host, it doesn't mean they're actually connected to each other, right? And that connection is critical. And so thanks for the link to that citation. We'll obviously take a look at it. And there are many other things that we're looking into in terms of improving the connection between the host and the passive pathogen.
Omar, just to add to what you have mentioned, if we have time in the end, I could demo an equivalent search in FungiDB, where essentially this is examining the same time points, but in Candida albicans.
Yeah, we probably won't have time just because we only have 10 more minutes in the webinar, and we still have Suzanne to go. And I'm going to skip the part I was going to do just for the sake of time. So we'll end it at that. There are many, but we will be covering this in future webinars, I think, in terms of the pathogen side of things.
You guys
go ahead. I think, Suzanne, you can go ahead, and I'll answer the next question by typing it out. So
okay. So yeah, 10 minutes. I'll do exactly what Eve said. That was my plan for an RNA-Seq experiment. So I'll just demo another RNA sequencing experiment and search in HostDB. And the data set that we're interested in is a mouse transcriptome during early and late infection with T. gondii. These guys, it's published by Garfoot, Cervantes, and Knoll in around 2019, and they took a bunch of samples, over a time course. They have 21, 28 days, all the way up to 6 months. They compare infected mice to uninfected mice, and then they also have some are looking at female versus male mice. They ran all of these samples in triplicate, then and deposited that in the sequence read archive, public data access. Then we accessed it from there and reanalyzed the raw data according to our standardized workflows, and including a statistical analysis on this. And we analyze all the raw data according to our workflows so that then everything on our site is comparable to anything else. And in our analysis, we aligned the reads to the mouse genome, and that data is in HostDB, and we aligned to T. gondii, and that data is in ToxoDB. So I'll demonstrate searches of this data in both HostDB and Toxo. It's kind of really kind of interesting. Just to save time, I'm already here at the Rna-Seq search page in HostDB, and I have to find my data sets. So I'm going to search for gondii. And I have 4 experiments to choose from, and here's the one from Garfoot that I had described, and I'm going to look at this in a statistical analysis. Previously, Eve had showed us how to run the fold change search, and this is I'll look at our statistical analysis. And so here I am at a search page. Again, I have the title and a bunch of parameters that I need to set up. For my reference sample, I want to look at the 28 days in female. For my comparator sample, I'm going to choose the 3-month sample from females, 90 days. I'll look for genes that are differentially expressed up or down, doesn't matter to me, however they are changing is good, with a fold change difference of 2 or more, with a adjusted p-value of 0.01 or less. Then I can click Get Answer, and my results then say that I have 651 genes that are differentially expressed between the 3-month sample and the 28-day sample. And these genes, right, are host genes, mouse genes that changed in response to infection with the T. gondii. And then, of course, within this system, you can go ahead and do all of our tricks and trades, add columns, add steps, download, run enrichment analysis, and whatnot. But the really interesting thing about this is that, there's a parasite component to that data too. And so I have switched over to ToxoDB, and I'm on the Rna-Seq evidence search here, and I'm going to look for this data. So I just filter data sets by mouse, and I find the Garfoot data set here again, and I'll choose our differential expression search, which is opening. I will choose as a reference, I'll set it up exactly the same. I'll choose to compare the reference with the 90-day sample. And you'll notice that we have fewer samples here, and that's because the uninfected controls don't contain parasites, right? So there's nothing in those that will align to the Toxoplasma gondii genome. So I've set this up to compare the 90-day sample to the 28-day sample, genes that are up or down, differentially expressed twofold or more at a just a p-value of 0.01, so same parameters, and I am returned 185 genes that are differentially expressed in the T. gondii parasite in response to infection in mouse. And I can then consider those alongside the host genes that are differentially expressed in these samples too, having run the same search in HostDB. Sound good? We have 5 more minutes.
Yeah, so one thing maybe if you can show, there was a question about downloading data, and maybe you can go ahead and click on the download link and just quickly show that.
Yep.
So just show where the download link is, yeah.
Yep, download link is there in the middle of the result page. Let me, should I go back because I wouldn't really.
Yep.
So I'm on my search result page. I have the strategy at the top, and down here in the list, in the table, there's a little link for download. These are the tools that you can perform on this set of results. And so if I click download, I'm taken to the our download tool, where I can choose any one of 5 different report styles. You can make a custom table. You can have a pre-configured table. I can get faster GF3. So I'll just go ahead and choose to make my own table. And then I can, any field that I searched in the text search, any data field I can add to this download. Let's say I want to look at my expression values for the Rna-Seq. I can drill down and look at those two. Click get genes. And then it might take a while. I didn't do this before, but then eventually I'll come up with a table of gene IDs for rows, and then columns are all of the data fields that I asked for. So it looks like it's loading now, right? So gene IDs on the left, and then many data fields on the right.
And the idea is that you can download this as a CSV formatted file, which you can open up in Excel. And if you want to graph this in a very different way, or you want to do different kinds of analyses on these that are not available in the database, you can do that. Mm-hmm. Great, thank you. So maybe we can switch to me so I can just show the final survey.
Yes.
So I did provide a link to our exit survey, and that's now in your chat box. You're feel free to also scan this box here. It's actually a very short survey. It's very quick to do, and please go ahead and do that now before before you leave, because if you don't do that, you're never going to do it. That's what we've learned. Make sure to visit our future webinars page. So if you go under Learn how to use ViewPathDB and go to Webinars, and this link is also in the chat box, you will see our future webinars that are scheduled. And next week, it's a webinar on building complex strategies. So some of the strategies that we built today, which are 1 or 2 steps or 3 steps, we're going to be doing more of those. We'll spend an entire webinar just adding steps and doing different things to the strategy to show you all the sort of the details, tips and tricks of how to build a more complex in silico experiment. Okay, I think this is it. I'm looking to see if there are any other questions. So again, if you think of a question later on that you did not ask during the webinar, click on the Contact Us link on any of our websites. So at the top of the page, you will see a Contact Us link. and let us know what you think. Provide suggestions. If you know of data sets that should be integrated, let us know about them using the Contact Us link, and ask us any question you want, and we'll be more happy to answer it. All right, well, thanks a lot, Eve and Suzanne, and everybody else who was helping behind the scenes, and hopefully we'll see you all next week. Take care, and bye-bye.
Bye-bye.
Bye.